Hello. Today we're going to do the M4 Universal Biology final review contents, and we will cover everything that you should know for the 2025 semester 2 final. So let's get started. So we have 3 sections to cover here. The first is from chapter 4.1 in the textbook, and that is organic and inorganic compounds and nucleic acids. Then we're going to look at chapters 7.6 to 7.9, the structure of the human gastrointestinal tract and digestion. And then finally, we will look at animal gastrointestinal tracts of 2 different types. So the first part from chapter 4.1, organic and inorganic compounds and the nucleic acids. We learned about 4 main types of biological molecules, and those are the carbohydrates, proteins, the lipids, and the nucleic acids, which make up DNA and RNA, the genetic material. So we will start with number 1, which are the carbohydrates. And the important thing to know here is that these serve as a source of energy for the body. They provide energy for metabolism, and they're very essential to life. We consume most of these as something called starch, which are the complex carbohydrates. And we break these down into the simple sugars, such as glucose, which we call simple carbohydrates. And there's a couple of other names for these, which we will look at a bit later. Namely, for the complex carbohydrates, they're called polysaccharides because they're made of many sugars, whereas the simple ones are called disaccharides if there's 2, and monosaccharides if there's just 1. We have to break down any carbohydrates that we eat into the simplest sugars so that they can be absorbed, because the larger carbohydrates cannot be absorbed through the cell membrane. Next section we want to look at are the proteins. So proteins are used by the body mainly for growth and repair. There are also many other functions for proteins, which are as enzymes or other specialized functions. So we'll look at some of those. But what you need to know about proteins is that they are composed of amino acids. The smallest unit that they break down into is amino acids. And when we eat protein, our body has to break that protein down into amino acids to make them readily absorbable, basically, so that they can go into the bloodstream. So proteins are broken down into amino acids, and starch and carbohydrates are broken down into the simple sugars. Let's skip ahead to number 4, the lipids. Lipids are fats and oils, and they act as a storage system. They act as a storage system for energy. Much as starch acts as a storage system in plants, these act as a storage system in animals. They are made up of glycerol and 3 fatty acids, and that is the simplest unit they can be broken down into before they get absorbed. So in our digestion, we have to break down lipids into glycerol and fatty acids. Lipids are extremely important in the construction of and maintenance of cell membranes because they are made of a bilayer, a 2-layered hydrophobic membrane that is what separates them from the surroundings and allows them to screen materials that go through them. So let's look at an example of a protein in number 3. Hemoglobin is the protein used to transport oxygen in the blood. It's what makes blood red. It's that red color in the red blood cells. So an extremely important protein in the body, hemoglobin. Let's look at another molecule here. So let's have a look at number 5, and then we're going to kind of skip ahead to 7, which is related. So those nucleic acids, one of the main molecules they make up is DNA. And DNA is our genetic material that is found in the nucleus of every cell. It's the control center of each cell, and it controls a lot of the functions of the cell. It controls the growth and structure of the cells. And throughout the body, it carries out different instructions, all from the same DNA that is found in each and every nucleus in every cell. And DNA stands for deoxyribonucleic acid. So do remember what it stands for. It's a short way of saying deoxyribonucleic acid. Let's move on to number 7, the structure of that DNA. How is it shaped? So unlike in bacteria, where DNA is either a long linear strand or it can be a circle called a plasmid. It forms a double strand, and the structure is a double helix, which means it is a spiraling coil. It's 2 spirals that go around each other like a spiral ladder. So that's the structure of DNA. I want to skip ahead to number 8. So number 8 is the term I mentioned before for the complex carbohydrates, polysaccharides. Polysaccharides are these large carbohydrate structures that are made up of many, many simple sugars put together. Examples of polysaccharides include glycogen in the human body, starch in plants, and cellulose in plants. So starch in plants, as I said, is used for storage of energy, whereas cellulose is actually the physical structure of the plant, the tissue of the plant. It's quite tough and durable. It makes up the cell walls of plant cells, and it gives them their rigid structure. It's also quite difficult to digest, which is a point we will come back to again in the animal digestion, the strategies that animals use to digest cellulose. So those are examples of polysaccharides. Make sure you know those. It's important to remember in number 9 that all organic molecules contain carbon and hydrogen. This is why we refer to life on Earth often as carbon-based. We don't know if there are other bases for life elsewhere, but here it's carbon-based. It always has carbon and hydrogen elements in it. We're going to go back to number 6 now, the process that breaks down large molecules in the body, which is called hydrolysis. And the key to the meaning of this word is in the name, hydro meaning water, and lysis meaning to break. So water break. Literally, water breaks these molecules down into smaller particles. Okay, moving on. One of the tests we did in the lab for lipids is called the emulsion test. That's the test where we put oil together with water and alcohol, and we ended up with 2 layers, one separate layer of oil on top of the layer of water. And that's due to the hydrophobic nature of lipids. It indicates that lipids are present And it's different from the color tests that we used for starch, where we used iodine, which will turn blue in the presence of starch. And the test that we did for simple sugars with the color range from blue to red, indicating the amount of simple sugars present. So the emulsion test indicates lipids. Let's go back to enzymes for a moment. The proteins that act as catalysts, and catalysts speed up any chemical reaction without being used up. So enzymes have special requirements in order to work. They have an optimum value of the certain factors such as the pH, how acidic or alkaline the environment is, or the temperature for that enzyme to work. It is their optimum range, the best temperature where they work the most efficiently. And then if you go outside of that, you have problems, because what happens is that you change the shape of the protein if the pH is too high or low, if the temperature is outside of the optimum range, then you can get a process called number 12's denaturation. An example of denaturation is when you cook an egg. You change the shape of the protein so much that you cannot change it back again. So just like you cannot uncook an egg, you cannot fix an enzyme after its shape has been denatured. So this is a change in the shape of an enzyme so that the active site cannot bind to the substrate anymore, the substrate being the substance that it reacts with, that it binds with in order to create a new products, basically. So that enzyme, those enzyme requirements, the optimum value, meaning the best value that it works in, is necessary to prevent this denaturation so that the enzyme can work. Enzymatic reactions, this is a diagram you need to know, you need to understand the different parts, the names of these parts, and to explain this reaction, I'm going to use an example that we did in the lab. That was the catalase experiment, where we took catalase enzyme from ground up raw potatoes, and we added hydrochloric acid to see if a reaction would occur. So there's a specific reason why catalase does this. Catalase breaks down hydrogen peroxide in the liver, which is a toxic substance, and it needs to be made harmless. So we have this enzyme called catalase. So we'll use that as an example here. So starting with number 1, that is called the substrate, the blue and orange objects together. This is the substrate. And in this case, let's use the example of hydrogen peroxide, which we did in the lab. If you look now at number 2, the red semicircle, you can see that it has specific indentations. Those are the active sites. And the substrate fits perfectly for only one type of substrate. So for catalase, it only fits perfectly with hydrogen peroxide, and it will only bind to hydrogen peroxide and react with hydrogen peroxide. So in that case, hydrogen peroxide is the substrate, and catalase is the enzyme. So what you need to know for the final is not the specific example in this case, but the general name for these parts of the reaction. Number 1 is the substrate, any chemical that is being acted on by an enzyme. And number 2 is the enzyme, which contains the active site and binds to a particular substrate. So then we move on to number 3. So number 3 is not a separate molecule. It is the same one. So once that substrate and enzyme bind together, you end up with one large molecule, which is called the enzyme substrate complex. That's number 3. That's the whole substrate bound together chemically with the enzyme. In this case, it's hydrogen peroxide and catalase. Now the actual reaction occurs, and the reaction for catalase is to break apart the hydrogen peroxide into water and oxygen. So in number 4, these new results basically are called products. So the hydrogen peroxide has now been broken apart. Let's say the blue one represents water and the orange one represents oxygen in this case. Those are the products, and they are immediately released. What happens to the enzyme now, the catalase in this case? Nothing. It stays exactly the same, and it is reused over and over again thousands and thousands of times. So what prevented this from working in the lab was when we added a lot of vinegar to the mixture. We dropped the pH outside of the optimum range, and we denatured our catalase so that it no longer worked. That means it changed the shape of the active site, so the hydrogen peroxide doesn't fit like a Lego block into it anymore, and then it will not bind, and so the reaction will not occur. And that's why we didn't get the white foam forming when we added the vinegar to this reaction. Okay, so let's go on. Okay, some other organic molecules and processes that you need to know are that fats are insoluble. This is something we mentioned earlier. The fact that they're insoluble in water is what makes them good for forming cell membranes. It means that they can separate out from the liquids around them and form these membranes. Cellulose is the polysaccharide we mentioned earlier. It's important to know it is made of simple sugars. It is not made of glycerol or fatty acid. It is not made of amino acids. It is made of simple sugars. Another enzyme you need to know is lactase. So lactase is the enzyme that removes milk sugars from milk. All babies have lactase, and many people lose this enzyme, the ability to produce this enzyme, as they grow. A lot of people lose it around the age of, say, 13 or 15. And then at that point, you may become lactose intolerant, which basically means you just don't produce lactase, which breaks down the milk sugar lactose into simple sugars. So you cannot digest the lactose sugar in milk. Okay, next one are the restriction enzymes, and these cut specific sequences from a DNA molecule. That's their only function. Their reaction is to cut over and over again at very specific sequences. Then we have pectinase. Pectinase clears pieces of tissue from fruit juices. It clears the difficult hard tissues called pectin. So pectinase, another enzyme. And finally, we have catalase. Catalase is the one we did in the lab. It's the one that breaks down hydrogen peroxide in liver cells in order to detoxify them and make sure that that very toxic and damaging chemical is not present in the liver where it could really do some damage. Okay, let's move on. Okay, we're going on to the next section now, and this is chapter 7.6 to 7.9. the structure of human gastrointestinal tracts and digestion. So we will look at a few processes and parts of the digestive system here. First definition you need to know is emulsification. This is the process in which bile converts fats from large globules into much smaller droplets. So bile is a special liquid. It's produced by the liver, and it specifically breaks down fats. And how it does this is through this process of emulsification. Why? Because large blobs of fat, these globules, are very difficult to digest. They don't have a lot of surface area for enzymes to work on. So they have to be broken down into much, much smaller little blobs, little drops of lipids in order to be digested. So this process is called emulsification. The next definition we need to know is jejunum. So the jejunum is part of your small intestine, and it's the organ where the products of digestion, meaning the fully broken down amino acids, the simple sugars, and the lipids, which have been broken down now into glycerol and fatty acids, undergo the most absorption. So at this point, basically the products, the final simplified products, are being absorbed into the bloodstream through the walls of the small intestine. And that happens in the section called the jejunum. The next organ we need to know is the gallbladder. So although the liver is the organ that produces bile, it does not store it. It's the gallbladder that stores bile. So the liver produces the bile, the gallbladder stores it, keeps it there, and then it slowly releases it into the small intestine in the amounts that are needed to break down fats. So this is very important because without this, we could not really break down fatty foods at all. There are some types of animals that cannot eat fat because they don't really have much of a gallbladder, and fat is indigestible to them. Okay, another definition you need to know is enamel. Enamel is the hardest substance in the body, much harder than bone, and it covers the teeth to protect them. It protects the structure of the teeth. So enamel is that hard covering on the teeth. And then finally, you need to know that the stomach produces hydrochloric acid and enzymes. No other part of the body is really doing this. The stomach has a special mucus lining and a special adaptation to protect it from the extremely strong hydrochloric acids that occur there. Those hydrochloric acids help to prevent infection because besides breaking down food, they also kill bacteria. Of course, though, once they move from the stomach to the small intestine, your body actually has to produce other substances which are bases in order to counteract the hydrochloric acid so that it doesn't burn your intestines. Your intestines are not protected the same way that your stomach is. So the digestive processes have a series of names, and they go in order from 1 to 7 here. It's important to understand that this is a particular type of digestive system, which is called monogastric. Monogastric means it is one directional, it's one way. It doesn't have any special loops or features that go back and around again. And humans are an example of that. Horses, which have something called the hind gut to digest their food, are another example of that. And we will come back to this later when we talk about animal digestion. So these are the stages of digestion, and we'll go through them from 1 to 7. Number 1 is ingestion. So nutrition basically starts when the food and drink is taken into the body through the mouth. This is taking a bite of food or a drink. Ingestion. In humans, this is in small bites. In some other animals like wolves and so on, it's very big bites. In something like a snake, it's completely different matter where the whole animal is taken in. So we're going to look at the human example here. So that's ingestion. Next, we have number 2, mechanical digestion. And this is where the teeth and tongue basically break down the food into small pieces without a chemical change to the food molecules. That's not completely true because your mouth does have salivary glands, which produce the enzyme amylase. We will have a look at that a little bit later. Amylase, of course, starts to break down complex carbohydrates into simple sugars as soon as you are chewing on them, and it begins that process. But it doesn't take it all the way through. So we are going to consider that that's not the beginning of chemical digestion here. It's mainly mechanical. In other animals like birds, because there's no teeth, they cannot chew, and they will grind up their food in other ways. For example, by swallowing stones and keeping them in an organ called the gizzard, where the stones actually grind up and do the mechanical digestion. But in humans, it's the teeth. Okay, and we have some specialized teeth for these purposes. The molars mainly are for crushing and processing food for the most part. And of course, the teeth are covered with that very hard enamel substance so that they don't wear down too easily from eating. Okay, so next we move on. So a process occurs once the food is well chewed up. It is called a bolus at this point. It's a ball of chewed up food mixed with saliva in your mouth. And your esophagus, the tube between your mouth and your stomach, then carries out this process called peristalsis, these rhythmic muscular waves that push the food bolus down to the stomach. So basically, in the stomach, enzymes and hydrochloric acid break down the large insoluble molecules in the food into small water-soluble molecules, and that is the chemical digestion. So that's occurring in the stomach. And remember that the stomach has those special adaptations to protect it from the hydrochloric acid. So once it's passed through the stomach, it moves on to the small intestine. And it goes through a few stages here where there's some continued chemical digestion. That's when the liver produces bile, the bile stored in the gallbladder, which is then released into the small intestine to emulsify the lipids and fats. And simultaneously, another organ called pancreas releases pancreatic fluid, which breaks down proteins. So all of this is now happening in the small intestine. And it continues that chemical digestion from the stomach. It also neutralizes the hydrochloric stomach acid with a very strong base. Next, we move on to number 4, absorption. So this is where that organ, the jejunum, a part of the small intestine, comes into play, because basically these very small digested food molecules can now cross the wall of the intestine into the bloodstream or the lymph. So what happens there is that there are countless billions of these tiny projections called villi, which increase the surface area. And through this very thin one-cell thick layer, those simplified food particles such as the glycerol and fatty acids, the amino acids from the protein, and the glucose, simple sugars from the carbohydrates, are all small enough to go through the cell membrane and into the bloodstream in one case and into the lymphatic system in the other. So this is the absorption, and this is where the digestive system kind of parts ways onto 2 different paths. So those nutrients now continue really out of the digestive system and into the bloodstream and lymph, into step 5. So it's still part of digestion, but it's not really part of the digestive organs because we're now in the bloodstream and the lymph. So that digested food is moved to the cells of the body all over the entire body through the bloodstream and the lymphatic system, where they become part of the cells and are used for energy, growth, and repair. And that happens in the lymphatic system for lipids, and it happens in the bloodstream for glucose and amino acids. They are carried in the plasma of the blood. Okay, and that all happens through the capillaries, both at the absorption site where it's picking up those nutrients, and then again at the assimilation site where those nutrients are being picked up from the bloodstream into the individual cells. Okay, the next stage now is where the rest of it goes, which is what doesn't go into the bloodstream and what can't be digested. And that is dietary fiber and all the other indigestible things that are too difficult to digest. Those pass on through the body in a process called egestion. And in the process of egestion, also a lot of water is reabsorbed into the body. Okay, and then finally, at a certain point, before the very end, some excretory materials, basically toxins and things, are added to these digested substances, and then they're expelled from the body, and that is called defecation. So that is the final step, number 7, defecation. And not all animals go through all these steps. For example, snakes do not have mechanical digestion. They swallow their food whole, and they have extremely strong, much stronger than ours, stomach acids, which digest the whole animal. Some other animals have very powerful digestive acids, such as crocodiles, vultures, and hyenas, and so on as well. So there are animals definitely that skip mechanical digestion. Okay, so know all of these stages, what they are, which order they occur in, the difference between ingestion, you can remember that, obviously, because the food is going into your mouth, and egestion is food that is on the way out, but not at the final stage yet. Okay, so let's go on. Okay, an important definition to remember is the alimentary canal. That whole digestive system is basically a one-way tube. And that is that whole pathway of the digestive tract, including all the organs from the mouth to the anus, such as esophagus, stomach, and the intestines. It doesn't include the liver, doesn't include the gallbladder or the pancreas. Those are outside of that circuit. They just release fluids into it in the small intestine. The stomach is in that pathway, though. And of course, once nutrients get into the bloodstream, that's the circulatory system and the lymphatic system. It's not, strictly speaking, it's not part of the alimentary canal. A canal is just a tube. So this is the tube that goes all the way through the body. So just remember that. Okay, so we talked about the jejunum and these little projections that increase the surface area, which are called the villi. Each villi is actually covered with even smaller projections called microvilli. But we're just going to look at one villi, or rather a villus, since it's a singular. And we're going to look at the different parts. And these are parts that you need to know for the final. So the first one, number 1, is the epithelium. This is almost kind of like a skin. It's the covering, and it is only 1 cell thick, so that it is a very short distance for absorption by active transport and by diffusion. And that's very important for rapid absorption. It's very similar to the 1 cell thick rule of the alveoli in the lungs, where it has to be only 1 cell thick so that oxygen and carbon dioxide can exchange. But in this case, it's not oxygen, right? We are talking about the food particles which are going to be picked up. Number two is a special type of cell which needs to be there to protect the whole villi and the whole surface of the intestines, and that is called a goblet cell. So a goblet is like an old style cup from medieval times. It's a big cup and it describes the shape of this cell. And what they do is they produce mucus, and that protects the gut lining, the inside of the intestine, against digestion by your body's own enzymes. So there's a lot of enzymes out there. They're quite harsh. These digestive enzymes, not necessarily acid, but normally they're digesting these food particles, and they could also just digest the cells of your intestine and damage them. So the mucus cells, these goblet cells, are extremely important. They produce a protective coating to prevent the digestive enzymes from basically prevent you from digesting yourself in some way. Okay, on to number three. These are the blood capillaries. So these capillaries, very similar to the ones in the alveoli in the lungs, and the ones around every cell that provide oxygen and nutrients. So these transport glucose from the carbohydrates that have been broken down, and the amino acids from the proteins that have been broken down. And you can see these are little nets and networks of tiny, tiny blood cells. So they pick up through that one cell thick layer these nutrients, the completely simplified nutrients of the simple sugars like glucose and the amino acids into the bloodstream. Next, we have something called the lacteal, and this is another structure which actually transports the fatty acids and glycerol, which are the products of the fat breakdown by the enzyme lipase. And what this is a little bit different, because this actually is specified only to go into the lymphatic system. So it's not going into the bloodstream, it's going into the lymphatic system to transport these fatty acids and glycerol from the broken down lipids. Okay, and then next, at the base of each of these villi, they are lined with these cells which produce digestive enzymes, such as maltase, which breaks down the sugar, maltose. So they're continuing to break down into the final stages. It's a digestive enzymes that break down the food particles to the smallest possible ones that can then be absorbed into either the bloodstream or the lymphatic system. And then finally, number 6. So these are just called glands in this case. They're glands, number 5. And then finally, we have the vein. In number 6, this delivers the absorbed products to the liver through the hepatic portal vein. So you don't need to know hepatic portal vein, but vein is good enough. And this is actually taking it to the liver to be further processed, basically filtered to get out any contaminants. Okay, so know all of these parts, know how they match up with their function and what they do, what they're for, what they are called. Okay, so let's go on. Okay, a few other definitions that you need to know are mastication. This is the scientific word for the mechanical digestion that occurs in the mouth, the process by which food is crushed and ground out by the teeth through chewing. So this is literally chewing. It's called mastication. Another word we mentioned before when we were talking about mechanical digestion as well is the peristalsis with regard to the esophagus. So these are the waves of muscular contractions that push a bolus, a chewed up ball of food, down from your mouth to the stomach. That's called peristalsis. So peristalsis is that that rhythmic wave of muscular contractions. And reverse peristalsis is what happens when someone vomits. It's going from the stomach up to the mouth. And once again, it's these rhythmic waves that push the food back up. We will see that this is actually a natural process in some animals, not in humans. Humans, it means that the person is sick, or it could also mean that there's something very wrong with the food. Sometimes poison or some toxic chemical is detected, which would cause reverse peristalsis. And it's a very good thing that we have this. Not all animals have reverse peristalsis. We luckily do, so it is possible if you eat something poisonous and you're able to throw up quickly enough, you may be okay at possibly. Okay. Then we have the enzyme we mentioned earlier, amylase, which is secreted by the salivary glands. Once again, it starts to break down complex carbohydrates into simpler sugars before they're processed further on. And then the goblet cells, which we mentioned already, remember that they produce mucus that protects the gut lining against digestion by the body's enzymes. So just remember that definition is important. Okay, so we're now moving on to our final section, the animal gastrointestinal tracts. And we're going to look at two different types here. So we mentioned earlier that the horse has a monogastric system. And what this means is it just goes in one direction. Horses have something called a hindgut to digest grass, which means they have an enlarged area of their gut, but it still only goes in one direction. The food goes in the mouth, it chews the food, it swallows it. So that's the ingestion and the mechanical digestion have occurred. And then it starts going through the stages of chemical digestion and so forth. This is a monogastric system. This is also what humans have, and you could see in the pathway that we followed through the digestive system, it goes from one step to the next, and it never really loops backward or goes in any other direction. It just goes in one direction. The other type of gastrointestinal tract is more complicated, and that is called ruminant digestion. So you watched a video on this in class and answered some questions on this. So you saw an example of this, and you see these type of animals all the time among farm animals, most commonly with cows. So ruminants means that they have an organ called a rumen. This is a chamber of the ruminant stomach, which has four chambers, where the fermentation of grasses occurs before digestion. And it's almost always the case that it's a grass-eating animal, because grass is extremely difficult to digest. It's full of that very tough carbohydrate tissue called cellulose, which is very difficult to break down and get nutrition out of. So they have to do a special process called rumination and fermentation. And what you will see cows doing is chewing their cud, where they literally bring up the food from the stomach to the mouth a second time, even a third time, and they chew it again. They put it through mechanical digestion, through mastication, basically, over and over again, and then it goes back down to the rumen. So this is where fermentation occurs. Fermentation is a process of basically a process of breaking down by enzymes and so on, but it's performed by bacteria and microbes that live inside of the rumen. And it's a slow process, so it has to bring it up again and ruminate over and over again before it can actually digest the food. So all of this fermentation and rumination process actually happens before any chemical digestion occurs. And this is the diagram that you should know for this with the 5 stages. So we're going to link these stages to the processes for ruminant digestion. And you can see in number 1, that's the esophagus, just like we have. But then it moves on to this very large chamber, number 2, called a rumen. And from the rumen, in the rumen, it's going to ferment with those bacteria and protozoans that help to break down the cellulose in the grasses. If you looked at this stage, it would just look like chopped up, mushy grass. It's still green. It's still not very well broken down. And that's the whole point. So from there, it's going to move into the next chamber, which is called the reticulum in number 3, that little side pocket. The purpose of the reticulum is actually to contract and push that food back into the esophagus. And from the esophagus, it goes through reverse peristalsis, back up to the mouth. It is masticated again. It's chewed all over again with mechanical digestion. And then it is swallowed again. So you have normal peristalsis down the esophagus, number 1, right back to number 2, the rumen, the fermentation chamber. And this can happen a few times. It's the reason you often see cows just chewing and not picking grass or biting grass, because they have to spend probably more time actually chewing and re-chewing and ruminating that grass than they do actually ingesting it. So then from there, it goes into the next chamber. Finally, the reticulum just releases it to the circular chamber that you see in number 4, which is called the omasum. And the omasum is going to absorb water, minerals, and some other substances, the electrolytes. And then from there, it's actually going to go to step 5, the albumasum. And in the albumasum, it's going to be undergoing the chemical digestion now from enzymes, from stomach acids, which are going to break down lipids and proteins as well. Okay, so, and then from there on, it's going to go into the small intestine, and the same, very similar processes as a monogastric animal, where it goes through absorption, assimilation, and then the egestion, and finally defecation, while the nutrients go along their path to be absorbed, be assimilated into the different cells where the materials are needed. So this is a much more complicated system than monogastric because it has multiple chambers, which is necessary for digesting grass. It's one of the methods for digesting grass. Horses don't use this, but of course, horses have to have a huge hind gut, so they have a different adaptation. But for cows, this is what works for them to digest grass. And it involves continuous swallowing, chewing, swallowing, fermentation, and then regurgitation, the basically reverse peristalsis, chewing once again, swallowing again, and so on. And this process is called ruminating, hence the name ruminant. Okay, so these are the parts you need to know, and these are the names you need to know, and you also need to know what's happening in each section. So we're going to go over that in the next one here. So these are the stages, once again, of the ruminant digestion. So starting with ingestion, the grass is eaten and swallowed, and it goes down the esophagus to the rumen. Okay, so let's have a look at this here. Okay, so it's eaten and swallowed. It goes down, it's eaten, it's chewed, it's masticated, chewed up, mechanical digestion is occurring, and it goes down the esophagus by peristalsis to the rumen, that big chamber. We'll go back to look at that in a minute. So it goes down, back down to that big chamber, the rumen, where it is fermented. So this is the next stage here after ingestion, is a fermentation. So the grass is fermented in the rumen. It is broken down by bacteria and microbes that live in the rumen. And then it moves on to the next small chamber, the reticulum. So let's go have a look at that just to remind ourselves what that looks like. There's number one, the esophagus. There's number two, the big reticulum, which you see does not go directly onto another chamber. It goes back up and then onto number three, the reticulum. And that reticulum contracts and pushes that grass, the half-digested grass or fermented grass, back up the esophagus to be chewed again. Okay, so once that's happened a few times, so it's actually going from ingestion to fermentation and then back to mechanical digestion again, back to fermentation, to the rumination, the reticulum contracts. So that's what's happening in number three. The reticulum contracts, it forces food back to the mouth where it's chewed again and then swallowed back to the rumen. And then you go back to fermentation again. So it goes back between fermentation to rumination, fermentation in the rumen, and then into the reticulum, back to the mouth, back to the rumen, and so on. And this can happen again and again until it is actually ready to go through the final stages of digestion. And that's what starts to happen in the next chamber. So finally, after this is cycled through a few times and it's ready, then the reticulum releases the several times ruminated and fermented grasses into that round chamber in number 4, which is called the omasum. And then the omasum, you get water absorption. So water absorption occurs there. That fermented grass moves into the omasum, and the water, electrolytes, and minerals are absorbed in that location. Okay, then finally, once the water has been absorbed, it moves on to the last chamber, the 4th chamber of the stomach, number 5, the albomasum. So we've got omasum for water, minerals, and electrolytes, and then we've got the albomasum for the final stages of digestion, where hydrochloric acid and digestive enzymes break down proteins, fats, and other molecules and ready them to go onto the small intestine, where absorption can finally begin. So this is a long process. It's not the most efficient process either. It takes a great deal of time. Cows and other large animals like a water buffalo and so on spend a huge amount of time eating grass and plant material. It's not very high energy. It's very tough. It contains a lot of cellulose. And so in their case, it requires this 4-chambered stomach to ruminate, to ferment it, to ruminate it again, chew on it again and again until it can pass on to the other 3 chambers of the stomach, from the reticulum to the omasum, where water absorption occurs and the electrolytes and minerals are absorbed, and then finally to the digestion, where the hydrochloric acid and the enzymes break down those final proteins and fats into amino acids, glycerols, and fatty acids. Okay. So what are ruminants? Ruminants, the definition is that they are these herbivorous mammals. They eat plants, and they have multi-chambered stomachs for fermenting and digesting tough plants such as grasses. It's almost always grasses because they're the toughest to digest. They contain a lot of cellulose. They contain a lot of silica as well. So next, we have this question about grasses. Are grasses difficult to digest? Yeah, they are extremely difficult to digest, very tough, and they require this fermentation and rumination process of chewing it over and over again, fermenting it, chewing it again, in order to break it down and just to get it ready for digestion. In the video, we also saw that to study these ruminants, such as cows, and in order to study the microbes inside them and the digestive process, scientists will put a device called a cannula, which is a small door into the animal's stomach. And they generally put this into the rumen. So they use this to study their digestion. And you saw this in the video where he has the kind of door in the side of the cow's digestive system. And we had a little bit of a chat about, like, does this actually hurt the cow or not? That's a very good question. Yes, it likely does to some extent. So the cannula provides this sort of door. They take the grass out and then they process it and check for what type of microbes are there and try to find ways to improve the efficiency of their digestion and so forth. Okay, so that's a cannula. Remember, it's a small door that is put into an animal's stomach. Of course, it is not naturally there. It has to be surgically done on the animal in order to study its digestion. So just to remember, horses are not ruminants. Horses also eat grass, but they have something called a hind gut, and they are monogastric. So don't confuse the way cows eat and water buffalo and so forth with how horses eat. Horses have a different process. They have a very large chamber at the back of the gut called the hind gut. So they actually do a different process where they digest everything as much as possible first, and then they let it ferment in the hind gut. But it only goes in one direction. Horses do not regurgitate the grasses to ruminate or to chew them again. They do not have a four-chambered stomach or anything like that. So quite a different process there. And in the end, it still works, but it's not a ruminant. So only animals with rumen are ruminants, and almost all of these animals are herbivores that eat grass. They're also quite closely related to each other. So that rumen and the four-chambered stomach evolved in one animal likely, and then it was that common ancestor that led to that digestive system being found in all of the others. Okay, so just to go back and review a bit, this is a ruminant digestion. For ruminants, the rumen is the organ that gives the name to ruminant. It goes through the esophagus, the rumen, the reticulum, the omasum, and the albomasum. Goes through this fermentation process again and again, through this rumination process, where it's going from the reticulum back to the esophagus, back to the mouth again, and then finally passing to the omasum. Digestion has not occurred yet. It's just absorbing water and electrolytes and so on at that point. And then finally, the digestion occurs in the albomasum with the hydrochloric acid and digestive enzymes. Okay, so that's basically that in a nutshell. And just remember that horses are not ruminants, cows are ruminants, buffalo are ruminants. And the reason that ruminants have this four-chambered stomach is due to the difficulty of digesting the very cellulose-rich grasses, extremely tough and difficult to break down. And remember, the definition of cannula is the small door into the animal's stomach that the scientists use to study their digestion. Okay, so that's all we have for today. Just make sure you know all parts of this and make sure that you know your diagrams. Make sure you can match all your labels. You should be able to describe an enzymatic reaction. You should be able to describe the rumination process and the process of digestion in ruminants. and you should be able to describe the different stages of digestion in humans and other monogastric animals. Okay, so this should cover everything. I will be posting the page references for the, not the page references, the question, which questions are from each section on your Google Classroom, and I will be posting the whole review package as well for you to have a look at on Google Classroom. Also, of course, if you have any questions, you can always ask me online or in the classroom, or you can come to the office and ask anything that you need. Okay, thank you. That's everything. And everyone, good luck. Oh, no, no. Oh, no, no, no.